A pleasant afternoon to everyone. First of all, I would like to thank the EMPEDA for inviting me to participate in this activity. RGCA EMPEDA is very close to my heart because I have worked with some of them for several years on the culture of the different phases of mangrove crab. So I'll talk about the various approaches that have been done to enhance the production of mangrove crab. We called that before mud crab, and now we propose to call it mangrove crab because it's more environment friendly. And based on FAO, it's called Indo-Pacific swamp crab. These are Scyila species, an important crustacean commodity next to shrimp. These are highly priced commodity in both local and export markets throughout the Indo-Pacific region. It's earning several dollars for a lot of countries because they export the commodity. Mangrove crab farming has become popular because of the repeated diseases that has been experienced in shrimp farms. And even if the crabs and shrimps share the same type of disease, because they're both crustaceans, crabs are sturdier, no? When infection gets into the pond, which commodity dies first? Shrimp, right? And so many people are now shifting to crabs. And aquaculture production based on FAO statistics in 2006, no, that was long years ago, the production was 13,792 tons. And after 10 years, it increased into 89,390 tons, valued at $796 million. And most of these seed stocks came from the wild, and only a few countries are getting the hatchery stock, no? Or the stocks for farming from the hatchery. Countries like Vietnam, Philippines, India, and then Fiji, no? And a few from Malaysia also. And now establishment of hatcheries are increasing. So several issues have been identified pertaining to the industry, and these are the lack of supplies, cannibalism, fish as feed, because they're using fish for the grow-out ponds, and fish, as we know, is also being consumed by human beings. And then diseases, especially in the grow-out ponds. And then molting, especially for the soft shell crab production. Synchronous molting is needed. And then species identification, especially in countries where more than 2 species of mangrove crabs are present. And different approaches were also done to address these problems. Phase by phase, like in the hatchery, improvement of the existing technologies, and then in the nursery, the same thing, because there have been some technologies established long before, and we still need to improve this so that the viability of producing mud crab becomes very high. And then in the grow-out phase, we have the development of immunostimulants, and then the refinement of feeds. And we also included here the post-harvest. And then we also included the genomics. This is now very important in aquaculture and resource management. And of course, diseases in all aspects of the culture phases. And all of these are towards to the production and sustainability of the industry. And whatever technologies are developed, then these are disseminated to the stakeholders or the users, no? Okay, so first we have developed the domestication of the mangrove crabs, and this is a prerequisite to selective breeding. We focused on the Scyila serrata because this is the species that the farmers prefer, especially in the Philippines, because they grow fast and less aggressive. And we have found out that you can close the cycle in 9 to 12 months. So this is now the production cycle, which was developed more than 5 years ago, and it's now, you can find this in the FAO fact sheet. We start from the sourcing of the broodstock, then to the hatchery, and then to the nursery, no, for the rearing of the small crabs that you produce from the hatchery, and then to the grow-out ponds, where you again get the mother crabs for the hatchery. Normally, the hatchery operators get the crabs from the trading centers or anywhere, like directly from the ponds or from the wild, and the quality is not very consistent, and so selective breeding here is very important, no, to improve the traits of the crab. So the project here involves the selection of Scyila serrata aimed at producing subsequent generations that exhibit resistance to disease and faster growth. So in the selective breeding that we started, we got only only selected 2 sites where the genetic diversity is very high. We found more than 17 alleles, no? And the size range of the mother crabs, 590 to 754 grams. And these were allowed to spawn in the hatchery. And this is the scheme that we use for the breeding. So we selected the normal growing crabs, or those crabs that most of them are in the middle, no? And then we also selected the fast growing, and we use this for further rearing. And then again, we selected the normal as the control, and then the fast growing as our treatment, no? And we followed the production protocol for this breeding or rearing the larvae. So we stock the, can you still hear me? It's so noisy. So we stock the larvae in the big tanks, no? This is a commercial scale production. So we produce the crablets as crab instar 1, 2 after about 23 to 25 days. And then these were placed in net cages in ponds for further rearing, because if the crab instars are immediately stuck in the grow-out ponds, you will get lesser survival rate. So we still put them in the nursery for adaptation, no? So that they can adapt better in a harsher environment when they go to the grow-out ponds. And so before we further rear them to grow out ponds, the animals were checked for WSSV, and only those animals that are free from the WSSB are used for the grow out ponds. And these animals were allowed to mate in the ponds. When the crabs are in the 150 grams, these were harvested and selected again. So stuck again in ponds for at 1:1 ratio, 1 male to every 1 female. And after 5 to 6 months, the crabs were harvested and then selected again for the broodstock phase, no? So the animals were placed in broodstock tanks for further maturation. And based on the selection, we have seen the improvement from the base stock or base population to the first generation. I think some of the rows are missing, but we were able to get up to the F2 generation, the second generation, and there was an improvement in the duration, rather in the range of zoea produced by 1 female, and also the duration from the zoea to the megalopa. It was shorter, no, in the first generation, and it became shorter as the generation is increased. And then the survival rate also is increased. So in the Philippines, if the survival rate from zoea 1 to C1 is about 3%, the hatchery owners can have profit already because the farmers, they buy the crab instar 1 or 2, no? Even if it's small in size, they can still be disposed. And so here in the pond, the growth also was heritable. So from the first generation to the next generation, you can see that there is improvement in the growth if you select the stock. But if you don't select, it's just normal. But if you select the animals to be stocked, those fast-growing animals, then you improve also the growth. And it came out that WSSB in the grow-out pond was not really our serious problem. We found this hematodinium. I don't know if some of the farmers have experienced this. This, on the top, you see the hematodinium infected crabs. And below, you see the normal crab. The crabs infected with hematodinium, the hematodinium is internal dinoflagellate parasite that causes bitter crab disease. This has been observed or experienced in other countries on blue swimming crab or other species of crabs. But in the Philippines, we just experienced this recently. And you can see the difference in the color. It's opaque, and then orange or reddish in color when you see the ventral side. And then the crabs are weak and lethargic. This is highly pathogenic, and it can wipe out the whole population of mud crabs in the pond. So now we are working on this, on how to prevent, okay? But it has something to do with your pond preparation. So we have already established the feeding and water management of crabs, and this is now the much improved version. Also, we feed them with the rotifers, 10 to 20, up to the zoea 5. There were many studies already that reported that the survival and growth is improved if you continue to feed the crabs with crab larvae with the rotifers. In Vietnam, they start feeding the crab larvae with the umbrella stage Artemia or the small strain Artemia. But we have tried this with this Scyila serrata. It doesn't work very well. In Vietnam, they are working on the Paramamosane, which is a little bit different from our, it's entirely different from our species. Okay, so here, we also feed the Artemia starting late soya 2 until the early megalopa stage. And then we also feed the artificial diets. Artificial diets are fed at starting soya 2 because the mouth parts of the crabs are more developed at that stage. Soya 1, not yet, based on so many studies. And then we feed, to reduce the amount of Artemia that we feed to the megalopa, we start feeding the mussel and fish, no? But very, very fine particles. And then we add microalgae, green microalgae, like the Nanochloropsis or Tetraselmis, because they shade they act as shade, and the water is improved, no? Because you know already that green water is very good for the animal. And water replacement is done every 3 to 5 days, depending on the quality of the water, about 30 to 60%. And we also put some prophylaxis here. Okay, so based on other studies, feeding of crab larvae with, this is more specific explanation based on the table that I have shown earlier. So feeding crab larvae with Artemia and rotifers enriched, with bacillus as probiotics resulted to better growth and survival. So the green water system is suitable for rearing the Zoya 1 to Megalopa and results increased survival and growth. So in other countries where importation of concentrated algae is not a problem, they are now using the concentrated algae, to reduce the culture of natural food. Excuse me. And my colleagues also did some experiment on the extracts of Terminalia catapa, potential probiont, Bacillus subtilis. This one is also found here in India. This is an effective antimicrobial and probiotic for crab larvae. So for the breeding, we use a different species of the worm. This is the Marfisa mosambica, which can be found in mangrove areas. Easier to culture than the marine annelids that we used to, like the Nereis. Excuse me. Nereis are more difficult to mass produce. So this one is easier. The life cycle can be closed in 2.5 to 3 months. Thank you. Yeah, thank you. And we use the marine annelids together with other natural food to improve the reproductive performance of the mother crabs. Ay, sorry. So we also did some quality assessment of crablets before we use them for grow-out ponds. So they should be active, of course, and then free of the white spot. And then, okay. And then no abnormalities, and then no pink color. Or no abnormalities like the albino. And then we also have this developed the type of shelter for the crablets. And then in the grow-out ponds, we have this formulated diets, and this is the basal ingredients or basal basal diet, and we found out that crude protein 48%, crude fat 5 to 6%, and the energy 1723 mj kg per diet were good for growing the crabs. And the crabs prefer water stable. They are messy eaters, so you need to have feeds that are stable in water, and then also crabs that they prefer spherical or cylindrical pellets, and a little bit rubbery, no? Because they hold the feeds using the claws. And so these are the other parameters needed. So cholesterol 0.17, then phosphorus 12.9, okay? And then here was also developed the stimulant for mangrove crab, ulva and sargassum. Different doses and frequencies were tested, and this came out, 1,000 milligrams per kilogram of diet can be used every 3 days, can be incorporated in the diet every 3 days, can be fed to the animals every 3 days, and then it can boost the immunological response and enhance the resistance of mangrove crab juveniles against WSSV. And this is now the novel, novel approaches. We integrated genomics with image analysis and geographic information systems, or GIS technology for improved rearing of mud crabs. We have, for easy identification, we just use these letters, no? Crab up, crab map, crab molt, crab adapt, and crab snip. Okay, so we have first the crabifier. In many countries, there are several species of crabs. No? So when they are small, it's very difficult to identify the crabs because small crabs are already stuck in the ponds. And we were able to develop this crabifier, a juvenile crab species identification, developed using DNA markers, image analysis, and mobile computing for the mangrove crab. So here are the different species. When they are still small, very difficult to identify. Like in the Philippines, we have more than 3 species, we have 3 species of crabs. And this one was tested. We collected a lot of crabs from different regions, and then identified first, confirmed first the identification, and then compared this with the apps that was developed. And here in the field first, it was tested, and then, rather in the lab first, it was tested, and then later in the field. And here, we use the frontal lobe spines for identification, and it works, no? You can download the apps. I think it's there in the abstract, the apps, the link to this application. And okay, so elevated water temperature in pond is one of the major problems in aquaculture. So here we also use the crab map. Crab map refers to temperature vulnerability maps for local major areas where mangrove crabs are farmed. So the ideal sites were mapped, and then identification of heat, stress, resilient stocks were also mapped, no? So what was the basis? It's the 30-year average temperature data at 1 square kilometer resolution that was downloaded from BioClim. So we used this and mapped the different areas in the country. The data was processed, no? And using QGIS to obtain the maps of temperature vulnerability. So we developed this site suitability map. Fish farmers can be guided as to which sites are ideal for their farming, and where also to collect the mangrove crabs that are more suitable for temperature adaptability. And we have also crab adapt, information on genetic adaptivity. This is related to the first application, okay? And then here, this is the map that was done to see the adaptation of the crabs. So the orange indicates adaptive crabs, moderately adaptive crabs. The red one, highly adaptive. And then the green, slightly adaptive, and then blue, least adaptive. So no highly adaptive in this area. This is only one example location, but there are also a lot of locations that we have mapped, no? So crab molt, another one, but this is more in-depth, no? Ratios of a molt promoting hormone to a molt inhibiting hormone were established for the different molt stages. But we also confirmed the stages using this conventional type where the legs are used to examine the molt cycle of the crab. And the crab molt is very important for those who are engaged in the soft shell crab production, so that they can control the molting of the stocks, no? So we have these crab snips, never mind. So the results of our R&D have been translated into improved production. And with these developments and refinements, we expect that production will be increased in the next 5 to 10 years. Okay, so